Long-term depression, limited, in neurophysiology, is an activity-dependent reduction in the efficacy of neuronal synapses lasting hours or longer following a long-patterned stimulus. Limited occurs in many areas of the CNS with varying mechanisms depending upon brain region and developmental progress. Limited in the hippocampus and cerebellum have been the best characterized, but there are other brain areas in which mechanisms of limited are understood. Limited has also been found to occur in different types of neurons that release various neurotransmitters. However, the most common neurotransmitter involved in limited is L-glutamate. L-glutamate acts on the N-methyl-D-aspartate receptors, NMDARS, alpha-amino-3-hydroxy-5-methylisoxazole 4-propionic acid receptors, AMPARs, kainate receptors, CARs, and metabotropic glutamate receptors, mGlurs. During limited it can result from strong synaptic stimulation, as occurs in the cerebellar Purkinje cells, or from persistent weak synaptic stimulation, as in the hippocampus. Long-term potentiation, LTP, is the opposing process to limited. It is the long-lasting increase of synaptic strength. In conjunction, limited and LTP are factors affecting neuronal synaptic plasticity. Limited is thought to result mainly from a decrease in postsynaptic receptor density, although a decrease in presynaptic neurotransmitter release may also play a role. Cerebellar limited has been hypothesized to be important for motor learning. However, it is likely that other plasticity mechanisms play a role as well. Hippocampal limited may be important for the clearing of old memory traces. Hippocampal/cortical limited can be dependent on NMDA receptors, metabotropic glutamate receptors, mGlur, or endocannabinoids. The result of the underlying limited molecular mechanism is the phosphorylation of AMPA-glutamate receptors and their elimination from the surface of the parallel fiber Purkinje cell, PCoFR8PC, synapse. Limited is one of several processes that serves to selectively weaken specific synapses in order to make constructive use of synaptic strengthening caused by LTP. This is necessary because, if allowed to continue increasing in strength, synapses would ultimately reach a ceiling level of efficiency, which would inhibit the encoding of new information. Long-term depression can be described as either homosynaptic plasticity or heterosynaptic plasticity. Homosynaptic limited is restricted to the individual synapse that is activated by a low-frequency stimulus. In other words, this form of limited is activity-dependent, because the events causing the synaptic weakening occur at the same synapse that is being activated. Homosynaptic limited is also associative in that it correlates the activation of the postsynaptic neuron with the firing of the presynaptic neuron. Heterosynaptic limited, in contrast, occurs at synapses that are not potentiated or are inactive. The weakening of a synapse is independent of the activity of the presynaptic or postsynaptic neurons as a result of the firing of a distinct modulatory interneuron. Thus, this form of limited impacts synapses nearby those receiving action potentials.